Hello everyone, my name is Maria Cristina Rumi and we are here at Georgia Tech today to discuss how to run a fluorescent measurement using a fluorimeter. So this is one such unit, it's a fluorimeter. This particular unit is from Horiba Jobeni Vaughan. Fluorimeters are composed of various units and they usually have a lamp. You can see sort of the light there will provide the excitation source for the measurement. Then there is a unit that is called an excitation monochromator. This is going to select the wavelength that is used for the measurement. This part is the sample compartment, and once we run the measurement we'll put our materials inside here. And the material fluoresces and the emitted light will be collected by this other part of the instrument, which is an emission monochromator, and then the signal is going to be sent to a detector that right here is sort of covered. In this case the detector is a PMT, it's a photomultiplier tube that is going to collect the photons. This particular detector is cooled. These hoses run water to keep the temperature of the detector down to improve the signal to noise of the instrument. The fluorimeter also has a computer and a data processing unit that collects the signal from the instrument and also sends a command to control the gratings and the slits, which are used to select how to run the measurement. And there is also a normal computer that is using an interface for the operator to instruct the instrument to do the measurement in a certain manner, and the monitor shows a sort of a typical screen before running a measurement on this machine. Fluorimeters are used to measure fluorescent spectra of material. This can be emission spectra or excitation spectra, and emission spectra can be used to determine quantum yields of fluorescence for materials. And today we are going to discuss how a quantum yield measurement is performed. We are going to discuss what is called the relative method. So we are going to measure one material with respect to another material that is known, that is being reported in the literature. And what we will want finally to do is to have all the quantities to fill in this equation. The quantum yield of a material is going to be the product of the quantum yield of the reference material, the ratio of fluorescent absorption properties, and we'll discuss those a little more in a moment, and then a factor that depends on the refractive index of the solvent in which the materials are dissolved. As for the fluorescence parameter here, I will show how to measure a fluorescence spectrum, and then we're going to obtain the integrated intensity of that emission. And then, as a separate measurement, we will need to determine the absorbance of the material at the excitation wavelength that is used to collect the emission spectrum. We'll need to prepare a solution of a material at concentration that is suitable for this type of measurement. The concentration is usually such that the absorbance of the material is relatively low. A typical number for the absorbance of a material is about 0.02. The reason for this is because we want the beam to be only very weakly attenuated when it passes through the material. And the reason is that we want the intensity to be as constant as possible, because we need to be able to compare the results of different materials, and the intensity of the fluorescence depends on the intensity of the light that is incident on the material. So if the absorbance is low, then the relative method can be applied. In particular, for this type of absorbance, the beam is only going to be attenuated about 2% along the path of the beam inside the cuvette. These schemes show a square cuvette with the incident beam and then the emitted fluorescence. It should be pointed out that the fluorescence is emitted everywhere. It's just that the detector is going to see only a portion of the light that is emitted. And this portion has, again, to be the same for the sample and the reference material that has been chosen. Now we are going to measure the fluorescence spectrum of a compound. I have here a solution of a dye in methanol. The concentration of the solution is relatively low, as we have mentioned before. The absorbance need to be approximately 0.02 or below. and this solution meets that criterion. So now I'm going to put this solution inside a cuvette. This quartz cuvette it has a sort of square cross-section of 1 centimeter each side, and it's made of good optical quality quartz. And I'm wearing gloves because I don't want to touch the windows on this material, because the oil from the fingers can damage over time the material. Okay, so now I'm going to take the solution and I'm going to put it inside the cuvette. So now I am going to close this and put the cap on the cuvette. Okay, so this is what we are going to use for our measurement. And one last thing before I put it inside the instrument, I want to clean the windows to make sure that there is no dust that is going to interfere with the measurement. So I'm going to take a little lens tissue and put some methanol on it. And I'm going to clean the window. And now we are ready for the measurement. We are now going to put our sample in the fluorimeter. I'm going to open the sample chamber and put the solution inside. And then we are going to close the lid, again, because we don't want light from the environment which is going to interfere with the measurement that we want to make. The fluorimeter lamp here is emitting light of all colors, and then the excitation monochromator is selecting one wavelength. In this case, we are using 350 nanometer light, and is sending it onto the sample. If I put a card in here, you can see that this is the light that is reaching our solution. And this yellow-green color that you see is the fluorescence emitted by the material. So now the instrument is going to collect that light at all wavelengths and show it on the computer. We are now going to start the measurement, and this is the interface that allows us to enter the parameters for the measurement in the computer. For this instrument and this software, we are going to, the menu is called Collect, and choose Experiment. We are going to have a window open up with various numbers. So we are going to select the excitation wavelength that we want to use for the measurement. In this case, it's 350 nanometer. We are going to have a range of emission wavelengths from 355 to 650 nanometer. We are going to integrate for 1 second in each wavelength. And we are also going to select the opening of the slits. In this case, we have about 2 nanometer bandpass on the excitation monochromator and 1 nanometer bandpass in the emission monochromator. These numbers are going to be different when you have a different sample, and you adjust them in order to have sufficient intensity to have a good signal-to-noise measurement. At this point, we enter a file name for the measurement, and we can start the acquisition. You can probably hear some noise coming from the machine while it moves various gratings to the position for the acquisition, and now you start seeing signal appearing on the screen. So we can see that we have 3 different graphs. The first graph is the signal that is collected by the photomultiplier tube, and this signal is proportional to the intensity of the light emitted by the sample, and is showed wavelength by wavelength. The acquisition occurs sequentially over time for each wavelength. We are also collecting in this second screen the intensity of the lamp that is exciting the material. And we do this to monitor if there are changes over time, and if we take then the ratio between this quantity and this quantity, we have our final measurement. It's the ratio between the signal intensity and the intensity of the light, and this is a number that we are going to use for the analysis. And the reason for obtaining the ratio between the signal intensity and the lamp intensity is to correct for possible fluctuations over time of the intensity of the light. Usually these are relatively small, but if you correct for this, you're only going to improve the quality of the data that you are collecting. And it's going to take 2 or 3 minutes to collect the measurement, because we have set 1 second integration time per wavelength, and we are going from about 350 nanometer to 600 nanometer. The measurement is now complete. We have collected the whole emission spectrum of our material. This is the sample that we wanted to investigate. You can see here the emission intensity as a function of wavelengths. At this point, we need to change the sample and measure the emission spectrum of the reference material for the quantum field measurement and repeat the same thing. For the reference material, we want the measurement collected under exactly the same conditions. So we are going to keep the same excitation wavelength and the same slit width for that material. Once we collect those two spectra and we collect the spectra of the solvent in which each one of those has been measured, we have all that we need to determine the quantum yield of the material. As I mentioned, we have selected 350 nanometer as the excitation wavelength. The choice of the excitation wavelength is done so that the material can absorb light at that wavelength. So 350 nanometer has to be within the absorption band of the compound. And you can see from the graph that in this case we are exciting somewhere on the absorption band. It doesn't have to be at the peak, but there has to be enough portion of molecules that are going to be excited for the fluorescence to be generated and have sufficient intensity for the measurements. We have now completed our measurement. We have here the display of the fluorescence spectrum of the sample compound that we have run and the emission spectrum of the reference material for the quantum yield measurement. At this point, we can export the data in usually in text format as column of numbers, where you have in one column the wavelength and the other one the intensity. And then you can use any data analysis software like Excel, Calligraph, or Origin to process the number and obtain the quantum yield results for this run. The information that is provided by the instrument need then to be processed to correct for certain effects. For example, the optical components in the instrument and the detector, the efficiency that depends on the wavelength, and so we need to correct for the response of the system. This is usually indeed called the correction of fluorescence spectra. It can be done often directly within the software that runs the instrument, or it can be done by the operator afterwards. Another thing that should be done is the subtraction of the contribution from solvents. These are usually relatively weak, but they should be measured and subtracted to obtain precise measurements. And this graph shows the spectrum as collected by the machine. The spectrum of the solvent is magnified 10 times to show that it is indeed weak. However, it has features, and in particular this band that you see corresponds to the Raman scattering due to the solvent. And then this curve is the response of the detector and the grating with wavelengths. So you can see that it is not a flat line. So we multiply the measured spectrum by the correction factor, and we obtain the corrected spectrum, which is the blue line. This is what we will use for determining the quantum yield. Usually the measurement is conducting on multiple concentrations of the same sample to have sort of repeat measurement, and then one way to process the results is to prepare a graph in which the intensity of the fluorescence and actually the integration and all the spectrum that I've shown before needs to be integrated, and the total intensity is plotted here as a function of the absorbance of the sample. The ratio between these two quantities was present in the equation that I showed at the beginning. So if you have multiple samples at different absorbances, you can fit the data with a straight line, and the slope of that line is going to be the number needed in that equation. So this is sort of one example of sort of the results can be obtained. Let's say that we have a sample and we have measured the fluorescence spectrum and the absorbance of the material. The absorbance is going to be measured by a different instrument, so it has to be done sort of separately. And the ratio of those two parameters is this number for sample number 1. And for the material that has been chosen as reference, the ratio of the fluorescent intensity and absorbance is this number. The samples and reference were dissolved in different solvents, so we need to consider the change in refractive index for the two materials. And we can insert the proper number in the equation that we have shown at the beginning, and in this particular case we are going to obtain a quantum yield of about 0.74. So this material has a fluorescent yield of 74%. As measured by this technique.